All right, so these are your notes on enzymes and chemical reactions. All right, so everything that your body does in terms of processing energy, um, doing dehydration synthesis, doing hydrolysis, all of those reactions are part of what's called metabolism. So you might say that, hey, I can eat pretty much whatever I want. I can have all the chocolate in the world, and I don't gain any weight because I have a high metabolism or I have a fast metabolism, right? That's what we kind of say in our everyday usage of the word metabolism. But what we really mean when we say that is that we have a lot of enzymes in our body that can quickly run and regulate chemical reactions. So anytime we're talking about metabolism or an organism's or a cell's metabolism, what we're talking about is the sum of all of the chemical reactions happening in that cell or in those cells that are breaking down materials or building them up. And those reactions are what is regulated by enzymes. And again, enzymes are just special proteins. All right, so a little bit of review from, I think, 8th grade or middle school. So reactants are anything in a chemical reaction that you start with. The word substrate is just a synonym for that. So whatever you start with could be called either the reactant or the substrate, or substrates. What you end up with is called the product, right? So if you have, and don't worry about balancing equations. We're not going to worry about doing any kind of math and making sure the numbers add up here. I know in middle school they probably had you do that quite a bit, but you don't have to worry about any of it for biology this year. But here's a quick example. If you take hydrogen gas and oxygen gas, then you can get water vapor. So hydrogen and oxygen are your reactants, those are your substrates, and then your product here is water, right? So when you run a reaction like this, typically if you just put the reactants next to each other, if you put the substrates next to each other, sometimes they'll react automatically, but many times, and at least in biology, most of the time they won't. You're going to kind of have to give them some help. And that help is called activation energy. You actually have to give your reactants a little bit of energy for them to start the reaction. So visually, if you want to think of this kind of like a metaphor, it's like kind of pushing a rock up a hill, right? Here are your substrates, but to complete the reaction, to move the rock to the right, you actually have to input or invest energy first, which is called the activation energy. And then once you've done that, the reaction will kind of go to completion and you'll get your products, right? Mathematically, the amount of activation energy is the height of this mountain or the height of this hill. So when we graph this, and I'm asking you, how much activation energy is needed, visually, if you're graphing this, so here's your y-axis and here's your x-axis. Your reaction might need, let's say that's 3 units of energy. You need to put in 3 units of energy before the reaction completes and makes a product, right? And we'll work on that in a little bit. All right, so again, enzymes are a type of protein and they are going to be the person, I guess, in this picture. If we go back, I guess this person would be the enzyme. They're the one actually making the reaction happen, right? So these are proteins that are going to help speed up reactions, right? Because most chemical reactions do happen pretty slowly. So the whole point of an enzyme is to make it go quicker, and they can happen thousands and thousands and thousands of times per second. Reactions are very, very quick. Probably quicker than you would be able to really imagine. Certainly quicker than you could count. The point, though, and the way they do this is to reduce high activation energies. They reduce the amount of energy you need to get the reaction started. So again, visually, in this kind of graph down here, it's kind of like lowering the size of the hill, right? So let's look at this red line. The red line would be the amount of energy you would need without an enzyme. So look at all this energy. Look at how much energy is used. You don't really need the number here, but all you need to see is just that it's bigger than the blue one, right? So you have to put all of this energy in without an enzyme, and then the reaction will move to completion, right? But with an enzyme in line B, you only have to put in, it looks like you only have to put in like half of that, right? You're only going up to there. So a lot less energy, and therefore it doesn't take nearly as much time to put that energy in, and the reaction can happen quicker. So this is how enzymes work. They make the reaction go quicker by reducing the activation energy. They lower the activation energy. And in this case, it goes just goes to show that you can build things in reactions. So here we actually have we have two substrates, A and B. Also, we have two reactants because those are synonyms. And in this case, we have one product. AB is the product. So let's look at how what enzymes look like. So another vocab word you need to know is that they are catalysts. Again, a catalyst is just anything that speeds up a reaction. So enzymes are catalysts. And they don't get used up. And that is a key feature of enzymes, is that you can reuse them over and over and over and over and over and over again. So let's imagine that kind of this little cartoony picture is an enzyme. So here we've got this lime green looking enzyme. And it's going to catalyze this reaction. So it's going to take two substrates. In this case, it looks like we're doing dehydration synthesis. We're combining these two substrates together, and we are getting one single product. And the key is that the enzyme didn't break, it didn't fall apart, it's still intact, and it can still work and do more enzymes over and over, or sorry, can do more reactions over and over and over and over again. All right, this is a 2D drawing, but like in your body, enzymes and substrates, they're all 3D, right? They all have the 3D space. So what we really care about is the portion of the enzyme that physically comes in contact with the substrates, right? That physically comes in contact with the substrates, and that is going to determine how they fit in together. If you look at the substrates, they look like they kind of fit into the enzyme. Right. So as we kind of progress through these notes, just keep in mind that the shape of the enzyme does match the shape of the substrates, and that shape is very important to the functioning of that enzyme, right? I know in the previous example, let me go back. In the previous example, we had two substrates and one product, but you can also have one substrate and two products. So in this case, the enzyme is doing hydrolysis, right? It's breaking down that substrate into two products. Up here in example 1, it looks like we're doing dehydration synthesis because we're putting two substrates together. All right, so again, talking about the shape of that enzyme, because that shape is so specific to a substrate, we call them substrate specific. So think of this like a lock and key. This is called the lock and key mechanism, right? One enzyme fits one substrate. That's it. It's not like any willy-nilly random substrate floating around the cytoplasm can fit in the enzyme. They are very, very picky, and they are very, very specific. So think of it like a lock and key, right? If you have a car, or if your parents have a car, you can't drive yet, but if your parents have a car, the car key is the one that fits in the lock, right? If you have a front door and your front door has a lock, the only thing that can open that lock is the front door key, right? You can't get into your house using the car key. Why? It's because the car key works with a car lock, and the house key works with a house lock. So it's the same thing with substrates and enzymes. They must match each other because the shape matches, right? Just like the key matches the lock, the substrate's shape matches the shape of the enzyme. Oh, and this picture on the right is kind of a computer version of what an enzyme really looks like. Remember, enzymes are thousands and thousands of amino acids all linked together. They're not these nice little teal blue blocks. They have complicated structures. So it's just kind of showing you the comparison. All right, so here's just another slide talking about the shape of that enzyme. The shape of the enzyme gives you what's called an active site. So the active site is the physical location on the enzyme itself that touches the substrate. So what I'm tracing right here on the left is what's called the active site, because this is the portion of the enzyme that physically comes into contact with the substrate, right? That's where the two are physically going to touch. That particular spot is the spot that cannot change shape. If you break an enzyme, perhaps by heating it up, and you break the active site, you change the shape of the active site, you've completely destroyed the enzyme. You've completely destroyed the enzyme. So down here in this second example, the active site is, again, where I'm tracing. I'm tracing over here on the bottom left. I'm tracing the active site in red. That is the spot that physically comes into contact with the substrates, right? I'm going to pause really quick, and I want you to focus on this picture right here, this middle picture on this top example, and this third picture on the bottom example. So when the enzyme and its substrates, all of the substrates, all of the components are completely together in one kind of giant unit, we call that the enzyme substrate complex. So again, when the enzyme is in full contact with all of its substrates before the products are formed, so the substrates have fit into the enzyme's active site, this entire kind of clump of molecules is called the enzyme substrate complex. Just some vocab for you to pay attention to. All right, so once this reaction is complete, again, this enzyme substrate complex, and it looks like this enzyme did dehydration synthesis because it took two separate substrates and linked them together into one product. And again, this enzyme can recycle. It's not broken, it can be reused, and it can just do this reaction over and over and over again as long as those substrates are present. Right? As long as those substrates are in the body, the enzyme can do this reaction. How do the substrates get into the active site of the enzyme, by the way? Right? You might have asked yourself this when you were looking over the video up to this point. How do they get there? Do they know how to get there? Are they being driven there by a chauffeur? a chauffeur? Think about it. Think about it. We'll address it towards the end of the video. All right, so other characteristics of enzymes. Just like you can destroy a lock on a door or a lock on a car, you can also destroy the active site of an enzyme, right? So when you do that, when you destroy the shape of the enzyme, particularly the active site, you've denatured the enzyme. That's the specific word we're going to use. We're not going to say it's broken. We're going to say it's denatured. And there are two big things that can denature an enzyme, that can break the enzyme. One of them is pH. If an environment's too acidic or too basic, that can denature the active site of the enzyme, and therefore it can just denature the enzyme itself, because that's the part of the enzyme that really works. The second thing that you can do is heat an enzyme up, and you can denature the enzyme that way. It's usually just heating it. It's high temperatures. Low temperatures tend to just slow the enzyme down. They don't actually denature anything. They don't break any bonds at the active site. Because again, low temperature is just low kinetic energy. So that's just slow movement, basically. So you might slow a reaction down with low temperatures, but you're not going to denature it with low temperatures. You're not going to denature the enzyme. So here's a quick example of enzymes with pH, right? You have enzymes in your digestive system because you consume food that needs to get broken down. So one enzyme that is in your stomach is called pepsin. And if you looked at the name, you might have been able to guess that pepsin breaks the peptide bonds in proteins. So pepsin breaks the peptide bonds in proteins. It frees the amino acids, right? Because those are the bonds that hold amino acids together. Well, that happens in the stomach. And your stomach has hydrochloric acid in it, and they have a pH of about 2, right? Whereas once you're out of the stomach and you've made your way into the small intestine, then you start to see other enzymes. So here's one called trypsin, right? Well, trypsin, since it's in the small intestine, functions in a much higher pH environment. The small intestine's pH is 8, right? There's no hydrochloric acid in there. It'll burn a hole through your intestines, right? Because the stomach has a lining that can handle it, but the small intestine doesn't. So if you took pepsin and put it in the small intestine, what would happen? Here's what would happen. It would denature. It would denature because pepsin is used to a pH of 2, right? This is the rate of the reaction. Look at how quickly pepsin is working at a pH of 2. It's its optimal pH, right? This is kind of the rate on the y-axis here. The higher up you are on the y-axis, the quicker the reaction goes, right? So pepsin loves working at a pH of 2. And it can even handle, it can go all the way down here almost to a pH of like 3.5. But after that, you've denatured pepsin. So if you put pepsin all the way over here at a pH of 8, it's super gone. It's super denatured by that point. You've destroyed it, right? Same thing with trypsin. Trypsin works really, really well. It has a high kind of rate of reaction at a pH of 8. It can handle, going up to 10. It's done at 10. That's as high as it can go. And it can go, maybe as low as 6. But it certainly couldn't be over down here at 2. Look how far away that is. You would completely denature trypsin. So pH 8 is way too basic for pepsin, and pH 2 is way too acidic for trypsin. They are unique to their kind of pH ranges, so they have a tolerance for pH, and if you move them out of that tolerance, they're going to denature. Here's temperature, right? So let's say, any enzyme in your body is going to function at your body temperature, right? I mean, if it didn't, then it wouldn't work. So in Celsius, we're just below 40 degrees Celsius body temperature, right? So all of your enzymes work at about 37 degrees Celsius. If you took one of your body enzymes and you put them in near boiling water, say over here around 100 degrees Celsius, they would completely denature, right? Interestingly, we have found bacteria that live in hot springs, naturally occurring hot kind of bodies of water that come out of the crust because they're getting heated by magma underneath the surface, right? Or lava I guess lava is once it's come out, and magma is when it's underneath the surface. So those bacteria, like the bacteria in Yellowstone National Park, where a lot of the geysers are, live in very, very hot water. If you drop this down where they function the best and you drop straight down to the x-axis, so you're looking at 70 plus degrees Celsius water. That's hot water. But because they've evolved in that environment, the enzymes that are part of the bacteria do just fine, right? So if you took those enzymes and you put it in a human body, well, a human body is way too cold. So the enzymes wouldn't react as quickly. They wouldn't denature because cold temperatures don't denature enzymes, they just slow them down. But they wouldn't work as well, right? But if you took a human enzyme and you plunked it down into a hot spring bacteria, you'd certainly denature that. Because our bodies don't go above, 40-something degrees Celsius. I mean, that's their range, right? Human enzymes can make it up to about 40, 45. What is that, about 50 maybe? And that's it. You put them over here in 90 plus degrees and they're done. They are denatured. right? So temperature and pH will denature enzymes. So here's an example. Here's another example. Peroxidase is an enzyme in your cells. If you ever cut your finger, or you have a little paper cut or something, maybe your animal or your dog or your cat scratches you, and you open, I'm not asking you to pry it open, but if you kind of like pull the skin a little bit taut so that you can kind of see into that, kind of pull apart the cut. If you take hydrogen peroxide, which is that kind of brown bottle that you can buy at CVS or any kind of over-the-counter drugstore. You can buy it at, Giant or Safeway or wherever. And you kind of dump it on your cut. And you can actually do this at home if you want. What that does is bubbles. It bubbles, right? Because in your cells, you have an enzyme called catalase. And it's going to take this substrate and it's going to break it down into two products. You're going to get water and you're going to get oxygen. So if you do that, and I'm not asking you to go get a paper cut, but if you do happen to have like a paper cut or something, just put a little hydrogen peroxide on it. And if you look really carefully, if the cut is new enough, you might even see some bubbles. So those are oxygen bubbles that are being formed. Proof that a chemical reaction is happening, right? So here's a quick little cute schematic of it. Here's catalase, the enzyme. Again, the active site is the portion of the enzyme that touches the substrate, right? So there's the active site. In this case, the substrate is hydrogen peroxide. And again, your cells naturally have catalase. It's part of every one of your cells. So this is the enzyme substrate complex, right? Everything is all kind of bound up together. This particular enzyme does a hydrolysis reaction, so it's going to break the substrate. And you get one substrate is water, and then the other substrate is oxygen gas. And this catalase enzyme can run this reaction again because they remain unchanged once this reaction is complete. Right. So hopefully you've had a second to think about the question we asked earlier. How do the substrates know, quote unquote, where the enzyme is? How do they meet the enzyme? There's no texting. They don't say, hey, meet me in the northern cytoplasm. They don't do any of that stuff. How do they find each other? Well, the answer is they find each other via random diffusion. Molecules in your cells and in your cytoplasm are constantly bouncing around and they are moving because they have kinetic energy, right? Temperature is just kinetic energy. It's movement. So as long as you have temperature, which I hope you do, even if you were dead, you would still have temperature. You're not at 0 degrees Kelvin. Molecules are always going to be bouncing around, right? And all it really is, and it really is this simple, is that molecules are diffusing around the cell. They're just moving and bouncing around and knocking into each other. And because there are so many molecules and there are so many enzymes and there are so many substrates, it's actually really, really common for them to hit the enzyme just by bouncing around. And that's actually what happens. And we'll talk plenty more about that in AP Bio. But they're not talking to each other. There's no communication here. There's no pre-planning. It really is just random diffusion and kinetic energy. So hope this helped, and I will see you guys next time.